In this video, we will discuss about prokaryotic DNA replication and the enzymes present in this and the steps included in this and the significance of this prokaryotic DNA replication. First, we want to know about the replication. What is replication? DNA replication is the process by which an organism duplicates its DNA into another copy that is passed on to daughter cells. So, let us consider this is the double-stranded DNA. Imagine that this is the double-stranded DNA from the parent. This will split into or they'll separate like this, and a complementary strand is formed both these strands. A complementary strand is formed for each strand. So we get structures like this. Here this is the new strand, and here this is the new strand, and this one is the parent strand. So both DNA contain one parent strand and one new strand, and this process is known as replication. That is, an organism duplicates its DNA into another copy and that is passed to the daughter cells. This process is known as replication, and this type of process is known as semi-conservative replication, which means half of them is conserved. Half of the DNA strand is conserved. So this type is known as semi-conservative replication because which contains one parent strand and one new strand. So once more, the DNA replication is the process by which an organism duplicates its DNA into another copy and that is passed on to the daughter cells. So these are the points. Replication occurs before a cell divides to ensure that both cells receive an exact copy of the parent's genetic material. Then replication uses a semi-conservative method, that is double-strand DNA with one parental strand, one new and a new daughter strand. And in prokaryotes, the model organism is mainly E. coli. But all the prokaryotes show many similarities with the E. coli. So the DNA replication is a fundamental genetic process and that is essential for cell growth and division. Now let us discuss about features of prokaryotic DNA replication. What are the features? First one is replication is bidirectional and originates at a single origin of replication. That is the replication starts at a particular position that is known as origin of replication. So in a double-stranded DNA, the replication starts at a specific position or particular position that is known as origin of replication. And the replication is bidirectional, which takes place in both directions. And the replication in prokaryotes takes place in the cell cytoplasm, and the synthesis occurs only in the 5' to 3' direction. And the individual strands of DNA are manufactured in different directions, producing a leading strand and a lagging strand. And the lagging strands are created by the production of small DNA fragments called Okazaki fragments that are eventually joined together. So these are the features. Once more, the replication is bidirectional and it originates as a single origin of replication. That is, in prokaryotes, the origin of replication only at one site. But in the case of eukaryotes, there are so many origin of replications. So in the prokaryotes, only one origin of replication, and that is known as orisi. and the replication takes place in the cell cytoplasm, and the replication occurs in the 5' to 3' direction, and the individual DNA strands, there are 2 strands in the double-stranded DNA. So one strand is synthesized in this direction and the other is synthesized in this direction, and the continuous strand is known as leading strand and the discontinuous strand is known as lagging strand, that we will discuss later. And the lagging strands are created by the production of small DNA fragments called Okazaki fragments, and they eventually join together. Now let us discuss about the enzymes used in the DNA replication. First one is helicases. Helicases are the enzyme which unwind the DNA helix at the start of the replication. That is, at the origin of replication or REC, the helicase binds and unwinds the DNA. Second one is SSB protein, that is single strand binding protein, which bind to the single strands of the unwound DNA to prevent the reformation of the DNA helix during replication. That is, the helicase, when the helicase unwinds the DNA, the SSB protein binds to each single strand. The helicase unwinds the DNA. At that time, the SSB protein binds to each strands of the DNA, which prevent the reformation of the DNA helix. The next one is primase. Primase, which synthesizes the RNA primer that's needed for the initiation of the DNA chain. So the primers are synthesized by the enzyme. Primers are synthesized by the enzyme primase. The next most important enzyme is DNA polymerase 3. DNA polymerase 3 elongates the DNA strands by adding deoxyribonucleotides to the 3' end of the chain. That is, the synthesis only occurs in the 5' to 3' direction because of the DNA polymerase 3. Then DNA polymerase 1, which replaces the RNA primer with the appropriate deoxyribonucleotides. These all we will discuss later. Then DNA topoisomerase 1, which relaxes the DNA helix during replication through creation of a nick in one of the DNA strand. So the nick is formed by the DNA topoisomerase 1 to relax the stress of the DNA. Then DNA topoisomerase 2, which relieves the strain on the DNA helix during replication by forming supercoils in the helix through the creation of nicks in both strands of the DNA. So, the DNA topoisomerase relieves the strains on the DNA helix during the replication by forming supercoils through the creation of the nicks in both strands of the DNA. The DNA topoisomerase 1 makes a nick only in one strand, but DNA topoisomerase 2 makes nick in both the strands of the DNA. Then the last one is DNA ligase, which forms a 3' 5' phosphodiester bond between the adjacent fragments of DNA. 3' 5' phosphodiester bond between the adjacent fragments of DNA. These are the important enzymes present in the DNA replication. Now let us look at the steps of DNA replication. Consider this is the double-stranded DNA. Here you can see this strand is in this direction, 5' to 3' direction, and this is in the 3' to 5' direction. So the direction is different in both strands. Now first of all, there will be an origin of replication. So let us consider this is the origin of replication. So in this origin, first the helicase enzyme binds. Helicase enzyme binds. That is, DNA replication begins at a specific site or spot on the DNA molecule called the origin of replication. At the origin, enzymes unwind the double helix, make its components accessible for replication. So the helix is unwound by the helicase enzyme to form a pair of replication forks. And the unwound helix is stabilized by SSB protein and DNA topoisomerases. Now, we can see that this is the origin of replication. Here the enzyme helicase will bind and it will unwind the DNA. So the DNA will be like this. That is, the hydrogen bonds here are broken down by the helicase. So it will be like this, the unwound DNA. In this unwound DNA, the SSB protein, the SSB protein, single strand binding protein, binds on each strand. The SSB protein binds on each strand, which prevents the rejoining of these two helixes, rejoining of these two strands. So SSB prevent the rejoining of the two strands of the DNA. And there are two enzymes, that is topoisomerase 1 and 2. Topoisomerase 1 and 2. When the helicase moves, it will unwind the DNA double helix. At the same time, a strain is formed on both sides of the DNA. So this strain is released by these two enzymes, topoisomerase 1 and topoisomerase 2. The topoisomerase 1 relaxes the DNA by giving a single strand nick, but the topoisomerase 2 relaxes the DNA by giving nick on both strands of the DNA. Nick on both strands of the DNA. So, first the helicase binds at the origin of replication, and when the helicase moves, it will breaks the hydrogen bonds between the strands, DNA strands, and the SSB protein, single strand binding proteins, binds to the each strands of the DNA, which prevent the rejoining of these two strands. Now we want a primer. We require a primer. Primer means an RNA fragment or maybe a DNA fragment which consists of 10 bases. And the primers are synthesized by the enzyme primase. The primase enzyme synthesizes the RNA primers, which consist of approximately 10 bases, which serves to initiate the synthesis of both leading and lagging strand. So here we can see that here two strands are there. One is in 5' to 3' direction. The next one is 3' to 5' direction. But our replication, DNA replication is always takes place in 5' to 3' direction. Always in the 5' to 3' direction. So first of all we want a primer. So the primase synthesizes a primer of 10 nucleotides. This is the primer. Here this strand is in the 5' to 3' direction, and here this is 3' to 5' direction. So the direction is like this, 5' to 3' in this strand. So the direction is like this, here in 5' to 3' direction. This is the direction. Here this is the direction. So the structure will be like this. Here we have one double stranded DNA which unwind by the enzyme helicase, and when the helicase moves, the SSB binds on both strands of the DNA to prevent the rejoining. Then the primer are synthesized by the enzyme primase, and this primer is added in the 5' to 3' direction. Here the direction is like this, and here is in this way. So the direction both strands are opposite, that is both strands are lie in anti-parallel direction. So the DNA polymerase only moves in the 5' to 3' direction. So in this strand the DNA synthesis is continuous, but in this strand it will be discontinuous. Let us discuss that the DNA polymerase 3, when the primer is added by the primase, the DNA polymerase 3 which add the nucleotides in the 5' end, 3' end. So it will continue like this. So the DNA polymerase add the nucleotides in the 5' to 3' direction. So when the helicase moves, it will continue the addition of the nucleotides. So in this case, the direction is like this. So the DNA polymerase add some nucleotides. Again, the RNA primer required. So this lagging strand requires so many primers, but the leading strand requires only one primer. So the lagging strand is discontinuous because the direction is opposite. It is a discontinuous strand. So when the primers add the primer, then that nucleotides added by the DNA polymerase 3, again the primase added another primer, then the nucleotides added by the DNA polymerase 3. In this alternate way, short fragments of DNA and primer are formed in the lagging strand. But in the case of leading strand, it is continuous. When we add one primer, then the nucleotides are added to this in a continuous manner. But in the case of lagging strand, the direction is opposite. That is, the direction is opposite in this case. So what we do? First add the DNA primer by the primase and adding the nucleotides by the enzyme DNA polymerase 3. Again added the primer by the primase, then added the nucleotides by the DNA polymerase 3, and this reaction is continuous in this strand. So we get a continuous leading strand, a discontinuous lagging strand. So the fragments formed in this, short fragments are formed in this strand, lagging strand. That short segments are known as Okazaki fragments. Okazaki fragments. Now we have a continuous strand and a discontinuous strand. Now we want to remove these primers, all these primers from the lagging strand. So here we can see this leading strand and one lagging strand. This is the lagging strand and this is the leading strand, this one. So now we want to remove these primers, and these primers are removed by the enzyme DNA polymerase 1. DNA polymerase 1, and the gaps are filled by the DNA polymerase 1. So these primers are removed by the DNA polymerase 1 and the gaps are filled by the DNA polymerase 1. So we get strands like this. This is the lagging strand and this is the leading strand. And the gap formed in this lagging strand and here is sealed by the enzyme DNA ligase. It is sealed by the enzyme ligase. So all the nicks are sealed and we get 2 new strands of DNA. Now we can check the points. The lagging strand synthesized discontinuously in the 5' to 3' direction through the formation of the Okazaki fragments. And DNA polymerase 1 removes the RNA primers and replaces the existing gap with the appropriate deoxynucleotides. Then the last one, DNA ligase seals the breaks between the Okazaki fragments as well as around the primers to form continuous strands. Now look at this figure. Here you can see a chromosome. Inside the chromosome there will be a DNA and here this is the origin of replication. This is the replication fork. So the replication fork is the Y-shaped structure formed during the replication. This Y-shaped structure formed during the replication is known as the replication fork. So here you can see this is the replication fork. Now in this you can also see the leading strand. This is the leading strand and this is the lagging strand synthesized in short segments, and this is the helicase. All these things we can see in this figure. And after the synthesis we get DNA like this. That is, this is the main DNA, then helicase unwinds the DNA and forming replication fork, and the DNA polymerases added the nucleotides and we get 2 new strands. At last that will be like this. This is the parent DNA and this is the new DNA. So we get DNA like this after the replication. So I repeat the process once more. First we have a double-stranded DNA. In this double-stranded DNA, in prokaryotes there will be one origin of replication. At the origin of replication, the helicase will bind. At the origin of replication, the helicase will bind and it unwinds the DNA. Helicase binds and unwinds the DNA, and following that the single strand binding protein, SSB protein, will come and bind to each strands of the DNA to prevent the rejoining of these 2 strands. Then the primer is synthesized by the enzyme primase. Primase means a short RNA or DNA segment, approximately 10 base pairs. So the direction is always in the 5' to 3' direction. The newly synthesized DNA is always in the 5' to 3' direction. So here we have 5' to 3' directions. Now the primase added the primer in the 5' to 3' direction, and the nucleotides are added by the enzyme DNA polymerase 3. DNA polymerase 3. The nucleotides are added by the DNA polymerase 3. So the one strand is continuous strand and the other one is discontinuous strand. One strand is continuous, other one is discontinuous strand. So the continuous strand is known as leading strand, the discontinuous strand is known as lagging strand, and the short segment of this discontinuous strand is known as Okazaki fragments, and these lagging strand require more than one primers, and these primers are removed by the enzyme DNA polymerase 1, and the gap are filled by the DNA polymerase 1, and the nick formed by the enzyme by this are replaced or sealed by the enzyme DNA ligase. These are the main processes in this replication. Similarly, two enzymes are there. One is topoisomerase 1, which relaxes the DNA by giving a single strand nick. Similarly, topoisomerase 2, which form double strand nick to relax the DNA during the replication. These are the overall processes. Now let us move to the proofreading. Proofreading means the correction of the errors. That is, in bacteria, all three DNA polymerases, polymerase 1, 2 and 3, have the ability to proofread using 3-5' exonuclease activity. When an incorrect base pair is recognized, DNA polymerases reverse its direction by one base pair of DNA and excise the mismatched bases. This process is known as proofreading. In bacteria, all the three DNA polymerases 1, 2 and 3 have the ability to proofread using the 3-5' exonuclease activity. So when they recognize an incorrect base pair, they will reverse its direction and then excise that mismatched base and add the correct one, then proceed. Now let us discuss the significance of replication. DNA replication is a fundamental genetic process that is essential for cell growth and division, and replication involves the generation of new molecule of nucleic acids, DNA, crucial for life. Then DNA replication is important for properly regulating the growth and division of cells, and it conserves the entire genome for the next generation. These are the significance of the replication. So after replication we get a new strand and an old strand like this. This is the overall process in the replication. Thank you.